Hello everyone, I'm Dr. Rahul Pai, lecturer in the Department of Conservative Dentistry and Endodontics, Yenepoya Dental College. For today's lecture, I'll be talking on a very important and a very vast topic in endodontics, that is endodontic microbiology. Robert Wayne once said, without microbial involvement in the pulp and associated periapical tissues, there will be no need for endodontic therapy. The ultimate goal of endodontic therapy is to prevent and treat apical periodontitis. Apical periodontitis is an infectious disease, and the rationale for endodontic treatment is to eradicate this infectious disease and prevent further microorganisms from infecting or reinfecting the root canal or other periapical tissues. A thorough understanding of the microbial aspects is essential for good endodontic practice based on a solid scientific foundation. Apical periodontitis as an infectious disease is an inflammatory disease of microbial etiology primarily caused by infection of the root canal system. There is scientific evidence to show that endodontic infection is essential for the progress and perpetuation of the different forms of apical periodontitis. Out of these microorganisms, bacteria is the major pathogen and others are fungi and viruses. Bacteria cause inflammatory and immunologic responses in the tooth structure and the surrounding tissues. This releases cytokines, MMPs, prostaglandins and rankel, which ultimately lead to connective tissue destruction, which shows clinical signs and symptoms of the disease in the host, and the disease progresses further on. For a brief history on endodontic microbiology, Anthony van Leeuwenhoek, once he invented the microscope, termed microorganisms as animalcules. W.D. Miller conducted a milestone study in dentistry. He found out the cariogenic bacteria that causes caries as well as apical periodontitis. Kakehashi in the year 1965 also conducted a very significant study with germ-free rats. Some other scientists who have conducted various studies are Sundquist, Moeller, Ricucci and Sequeira recently in the year 2010, where they described the formation of biofilms and termed the concept as community as a pathogen for the infectious role of the disease. There are various routes of infection through which microorganisms can gain entry into the pulp and the periradicular spaces. The tooth, as we know, is covered by an overlying cementum in the crown structure and overlying enamel in the crown structure and cementum in the root structure. This protects the pulpodentine complex from within. But any breach in the integrity to these layers, such as caries, trauma, cracks, fractures, any iatrogenic errors during the procedure, scaling and root planing, attrition, abrasion, or any natural gaps in cementum can lead to the exposure to the oral environment. And this provides a pathway for the bacteria to the pulpodentine complex. The bacteria can be from the caries, saliva, plaque, periodontal disease, etc. There is also another controversial theory called the theory of anachoresis. This theory states that bacteria can be transported into the blood or lymph to an area of tissue damage and establish an infection. However, there is no clear evidence that proves this theory. There are various articles and authors who have stated that there is no sign of transportation of bacteria from the blood system to the pulp system. Now, how does the vitality of the pulp help? As we all know, a vital pulp has various defense mechanisms. So, whenever there is infection by any bacteria, the initial stages of infection, there is outward movement of the dentinal fluid from the tubules. And the tubules also contain various components such as odontoblastic processes, collagen fibrils, lamina limitans. Hence, the functional diameter of each tubule is reduced to only around 5 to 10% of its actual anatomic diameter. And other factors such as age-related changes in the dentine like sclerotic dentine, tertiary dentine, smear layer and fibrinogen deposition also help in blocking off the tubules and preventing the further entry of microorganisms. Dentinal fluid also contains antibodies and complement systems which help in fighting off the infection. Hence, when the vitality is compromised, the amount of bacteria increases and this increased bacteria leads to a significant infection in the pulpodentine complex. Can endodontic treatment also serve as a route for infection? Yes. Microbial invasion during instrumentation or during inter-appointments or after obturation could be a cause of endodontic infection. During treatment, residual plaque, calculus, caries, leaky rubber dam, and contaminated instruments or irrigants can provide an entry for the bacteria. Inter-appointments due to leaky temporary restorations, fracture or loss of temporary restoration, or fracture of tooth may cause an influx of bacteria. And after the completion, that is post-obturation, a less than adequate coronal seal, recurrent decay around the sealed tooth, and fracture of the post-endo restoration can all lead to reinfection. Now that we've learned about the various infections of the pulpodentine complex, we should also learn what are the causative organisms that are behind this. There are more than 700 species in the oral cavity, and all have the ability to invade the pulp after the necrosis of the pulp. Modern culture techniques and molecular biology have helped us predict the organisms that are the causative organisms of the infection, and this has been shown to be polymicrobial in nature. And bacterial profiles of endodontic microflora may vary between individuals. These multi-species biofilm communities have been shown to have microcolonies irreversibly attached to the tooth structures and any interface that it comes in contact with. There are certain classification of microorganisms that cause endodontic infection. Mainly, there are anaerobic gram-negative bacteria, example Treponema, Dialister, Porphyromonas, Prevotella, etc. There are facultative gram-negative bacteria like Neisseria, Capnocytophaga. There are anaerobic gram-positive bacteria like Eubacterium, Actinomyces, Filifactor. And there are also facultative gram-positive bacteria, very commonly known as Streptococcus, Enterococcus, Actinomyces, and Lactobacillus. Now that we know a few microorganisms that cause the infection, we should also know what are the factors that lead to an infection. This is mainly known as pathogenicity and virulence. Pathogenicity is the ability of the microorganism to cause the disease, whereas virulence is the quantitative measure of the pathogenicity of a microorganism. There are various virulence factors. This can be microbial products, structural components, or strategies that contribute to pathogenicity. And also there are two types of pathogens, primary pathogens and opportunistic pathogens. Primary pathogens are the first pioneer species that cause the disease in the given host, whereas opportunistic pathogens are commensal bacteria that exploit changes in the balance in the host-bacteria relationship. Coming to the pathogenesis of apical periodontitis, bacteria are usually present in the early stages of pulp inflammation and necrosis. These bacteria later gain entry into the root canal space after the pulpal necrosis. Then they diffuse their bacterial byproducts, and these bacterial byproducts clash with the pulp and ultimately lead to the death of the pulp, leading to necrosis. The bacteria can move through the entire length of the root canal, and these are mainly the pioneer species. They later modify the environment within the root canal system. So there is a shift in the microbiota. It is more structurally organized as well as spatially oriented. Once this is done, the environmental factors such as oxygen tension, nutrition availability will determine whether the new species entering the root canal will succeed or will die off. We spoke about virulence factors, and these are the main factors that, by the way which bacteria exert their pathogenicity. They wreak havoc on the host tissues through direct or indirect mechanisms. Direct damage is mainly done with factors such as enzymes, exotoxins, and metabolic end products. Whereas indirect damage are considered to be more significant. Indirect damage is basically the bone resorption or the pus formation that occurs in the periradicular region. Indirect damage is mainly caused by factors such as lipopolysaccharide, lipoteichoic acid, peptidoglycan, lipoproteins, membrane proteins, DNA, and antigens. Now that we learned about pathogenicity, virulence factors, and the various microorganisms that are behind these, we should also learn about the types of infections that are present in the endodontic system. These are mainly intraradicular and extraradicular. In intraradicular infections, we have primary intraradicular infection, which is mainly caused by the microorganisms that initially invaded and colonized the necrotic pulp. These are usually dominated by the anaerobic species, and they consist of Tannella, Dialister, Actinomyces, Streptococci, etc. Whereas secondary intraradicular infections are caused by microbes that were introduced during treatment. They may be introduced between appointments or may be introduced after treatment. Common species are Pseudomonas aeruginosa, Staphylococcus, Escherichia coli, Candida, and other microorganisms. Persistent intraradicular infections, on the other hand, are caused by microorganisms that resisted the intracanal antimicrobial procedures such as cleaning and shaping, intracanal medicaments, etc. This is because these microorganisms can endure nutritional deprivation. E. faecalis is one of the most common culprits for persistent intraradicular infections. Some other microorganisms that cause are Candida species, Dialister, Streptococcus species, Tannella forsythia, Campylobacter, etc. Now that we spoke about the intraradicular infections, what exactly is an extraradicular infection? An extraradicular infection is basically formed when the microbial invasion of the periradicular tissue occurs. This is actually a sequel to intraradicular infection. Example could be an acute alveolar abscess. But sometimes these extraradicular infections may be independent, such as those infections caused by Actinomyces species. But this is quite rare. We spoke about persistent intraradicular infections and the role of E. faecalis. So let's dwell a little bit further into this very important microorganism in endodontics. E. faecalis is shown to be a major role in persistent lesions. They are common in root canal failure cases and are commonly associated with asymptomatic cases. This is because the E. faecalis can enter a VBNC state. What is VBNC? It stands for viable but non-cultivable. So basically, it can survive in unfavorable conditions and progress the infection only when the favorable conditions are present. Persistent and secondary infections are usually clinically indistinguishable, and they all may have persistent exudation, symptoms, intra-appointment flare-ups, etc. So what are the factors of E. faecalis that prevent it from being eradicated from the root canal system during our regular endodontic therapy? E. faecalis can endure prolonged periods of nutritional deprivation. As I said, VBNC state can be entered. It binds to the dentine and proficiently invades the dentinal tubules into greater depths than regular microorganisms. It alters the host response and prevents the eradication. It suppresses the action of lymphocytes. It possesses various virulence factors such as lytic enzymes, cytolysin, lipoteichoic acid, pheromones, etc. It utilizes the fluid present in the dentinal tubules or serum as its nutritional source. And it can also resist intracanal medicaments like calcium hydroxide. This is because of the buffer system that is present within the E. faecalis and also the properties of the dentine, which can reduce the effect of the calcium hydroxide that is placed in the canal. The main factor is that E. faecalis always forms a biofilm, which is more resistant to regular treatment. Now, what exactly happens once the microbial flora is introduced into the root canal? The root canal with a necrotic pulp has a warm, moist, nutritious anaerobic environment, and this favors the growth of microorganisms. Factors such as oxygen tension, available nutrients, and bacterial interactions further progress the growth of the microorganisms. We all know the root canal infection is a dynamic process, and at different stages of the infection, there are different bacterial species. Usually during the initial stages, facultative bacteria predominate, whereas in the later stages, obligate anaerobes predominate. As you can see here, the gradient of the infection from the coronal to the apical region varies. In the coronal region, there is high oxygen tension and you have good enough of nutrients from the oral cavity. Hence, there are higher bacterial counts, especially anaerobic bacteria. whereas in the apical region there is lower oxygen tension and lesser nutrients that are available. So the bacterial counts are pretty low, but there are very resistant bacteria in this area and especially anaerobic ones. Now that we've learnt a few things about how a bacteria survives and functions in an endodontic infection, we have to dwell further into a biofilm concept that has been recently introduced. It is called community as a pathogen. Individual microorganisms group to form populations, which in turn form microcolonies, and these microcolonies interact with each other to form communities. The infected root canal has shown to have a mixed community of microorganisms. The community and habitat are a part of a larger system, and this can be called the ecosystem. The biofilm can be defined as a sessile multicellular microbial community characterized by cells that are firmly attached to the surface and enmeshed in a self-produced matrix of extracellular polymeric substance or EPS. Now, why is this so important, and what exactly happens in the biofilm? A biofilm is a media for the bacteria for digestion, regeneration, exchange of information, exchange of nutrients, adherence to the structure, resistance to antimicrobial agents, protection from external sources, and quorum sensing. This is all possible because of the EPS matrix that is present in the biofilm, which contains proteins, polysaccharides, ions, channels, lipids, nutrients, and other cells and electrons that are present. The ability to form biofilms is also considered as a virulence factor. Microcolonies are usually 15% in volume, whereas the EPS consists of 85% of volume of the biofilm. These biofilms can reach up to 300 or more cell layers thick, and multi-species biofilm communities result in increased pathogenic effects on the host. The EPS matrix also confirms unique features to the biofilm community and plays an essential role in biofilm physiology, output, and protection. What are the advantages of this community lifestyle of the bacteria? They have a broader habitat range for growth of more diverse microbiota. There is increased metabolic diversity and efficiency due to food webs. More nutrition exchange can occur, and it also facilitates genetic exchange, which may be involved in the formation of antibiotic resistance within the biofilm. There is also protection from competing microorganisms, host defenses, antimicrobial agents, and environmental stress. Overall, there is an enhanced pathogenicity of the biofilm due to this community lifestyle. According to Caldwell et al., the biofilm has 4 following attributes: autopoiesis, or the ability to self-organize; homeostasis, or the ability to resist environmental disturbances and stresses; synergy, that is, the biofilms are effective in association with further microorganisms than in isolation; and communality, that is, the biofilm responds to the environmental challenges as a combined unit. Biofilms also have a classification, and based on the area that it is present, it can be classified as intracanal, extraradicular, and periapical biofilms. Intracanal biofilms, as the name suggests, is within the root canal system. Extraradicular biofilms are those root surface biofilms that form on the cementum around the root apex of an endodontically infected tooth. Whereas periapical microbial biofilms are isolated biofilms which are independent from the internal or external surface of the root canal. Now that we learned a lot about the biofilm and the community as a pathogen and the community lifestyle that the biofilm of microorganism adapts, we need to know a little bit more on the communication between the bacteria that occur within the biofilm. This is known as quorum sensing or the bacterial talk. It is basically the intracellular communication phenomena that is seen in a biofilm, and it involves the production and release and subsequent detection of chemical molecules. These chemical molecules are called autoinducers. In the initial stages, during the increase in the population of the microorganisms in the biofilm, the autoinducers produced also increases. Once the increase has reached a threshold level, the bacteria can perceive this threshold level, and the bacteria alters its gene expression. This is termed as quorum, or a balance between the threshold and the expression of the genes or the virulence factors. This regulates the virulence factors, metabolites and the biofilm formation for the further spread of the infection. Intraradicular biofilms usually are present in 80% of primary and post-treatment infections. This is mainly due to the invasion of dentinal tubules and the biofilms in the ramifications, lateral canals and isthmuses. Biofilms are more frequent in the case of a large periapical lesion, and the prevalence of intraradicular biofilms range from around 69.5% in granulomas to around 95% in apical cysts. Extraradicular biofilms, on the other hand, are only present in 6% of the recorded cases. Now that we've learnt about the biofilm, the bacteria, about how the infection progresses, it is also very essential for us to know about the methods for microbial identification. There are various methods. We'll be mainly talking about two, that is, the culture methods and the molecular biology methods. Some other methods that are used are microscope and immunological methods. Coming to culture methods, it is the process of propagating microorganisms in the laboratory by providing them with the required nutrients and proper physiochemical conditions, including temperature, moisture, atmosphere, salt concentration and pH. It is used to detect the etiology of apical periodontitis and for the composition of the endodontic microbiota. The steps involved in culture methods are the sample collection and transport, dispersion, dilution of the sample, cultivation of the bacteria, isolation of the bacteria and later identification of individual species. The media most commonly used are brain heart infusion broth, trypticase soy broth and glucose ascites broth. The bacteria are identified on the basis of multiple phenotype based aspects such as colony morphology, gram staining pattern, oxygen tolerance, comprehensive biochemical characterization, metabolic end product analysis, etc. There are certain advantages and disadvantages to this identification method. The advantages would be that it is broad range in nature, it allows for quantification of the bacteria, it is widely and easily available, and there is determination of the antimicrobial susceptibility of the isolates. Also, physiologic and pathologic changes are possible. Whereas the limitations are that it is technique sensitive. Not all viable bacteria can be recovered by these methods. It has low sensitivity and there is strict dependence on the mode of transport. Also, the fact that it is costly and time consuming and laborious can be a major drawback. Latest studies have shown that the culture of root canals prior to obturation is no longer recommended in clinical practice. This is due to the fact that there is no significant difference between cases that have negative culture prior to obturation and those that had no culture prior to obturation. These culture methods are sensitive and time consuming, and they can also always be sampling errors that are present. Hence, better cleaning and shaping techniques and understanding of the pulporadicular pathophysiology is more important and will lead to more predictable endodontics. Another method that is of significance is molecular biology methods. These are based on the identification of specific biologic markers present in the genes of microorganisms that aid in precise phylogenetic classification and identification of the microorganisms. Some commonly used molecular biology methods are PCR or the polymerase chain reaction, DNA-DNA hybridization and the FISH protocol. The PCR method is 10 to 100 times more sensitive than your culture methods. The 16S RNA gene has been identified as the most widely used gene for bacterial identification. The FISH method, also known as the fluorescence in situ hybridization, measures the abundance of target species and provides information on their spatial distribution in tissues. Molecular biology methods can identify strains with ambiguous and aberrant phenotypic behavior and rare isolates as well. Also, it can identify poorly described or uncharacterized bacteria and also identify new species. Coming to the advantages and disadvantages of this method, the advantages are that it is able to detect both cultivable and as yet uncultivated species. It has high specificity and high sensitivity, and it is a rapid method. It can also be used for antimicrobial treatments, and also it can detect the dead microorganisms. The certain limitations are that it is more qualitative or semi-quantitative in nature. It mostly detects only one species or few different species at a time, so it can be time consuming. It is a very costly method and some assays can be laborious. In conclusion to this topic, the philosophy remains unchanged in the light of new and convincing endodontic microbial research. The apparent presence of microorganisms in a canal does not ensure endodontic failure, nor does the apparent absence of microorganisms guarantee endodontic success. However, the presence of microorganisms, particularly certain types such as E. faecalis, provides an additional source of irritation that the body must overcome to gain optimum results. Therefore, we can say that the control of microorganisms and possible substances must be an objective in endodontic treatment. Thank you.